Chapter 3 on cells and tissues is a long chapter. Cells are the smallest living unit. Once we determined that cells existed, people started looking at all forms of life, and they determined that cells are the building block of all plants and animals. And this, as well as other statements, became part of what's called the cell theory. Cells are the building block of all life. All new cells come from pre-existing cells, and cells are the smallest living unit to perform all functions. All cells in the body have descended from a single fertilized ovum, the zygote. And from that zygote, basically 4 types of tissue have formed. We need to know all those 4 types of tissues and examples of them, where they are in the human body. Epithelial tissue, connective tissue, muscle tissue, and neural tissue. Cells do not live with just other cells. There's a lot of stuff between cells and around cells within tissues. Cells are always surrounded by fluid called extracellular fluid. When that extracellular fluid is between tissue cells, it's called interstitial fluid. And fluid inside cells is intracellular fluid. So extracellular fluid means outside the cell. Intra means within the cell. And of course, that cell membrane made of phospholipids separates the intra and extracellular fluids. Each cell is surrounded by that phospholipid plasma membrane. Within the cell, there is cytoplasm between the membrane and the nuclear membrane. And in the cytoplasm will be cytosol, the intracellular fluid, and organelles. This chapter separates organelles into membranous organelles and non-membranous organelles, but I'm not too concerned with that division. Here you can see the plasma membrane in pink, cytoplasm that consists of the cytosol and organelles, and then the different membranelles, or the different membranes and organelles. So in terms of organelles, know a brief phrase to remember what they do. Peroxisomes. Think of hydrogen peroxide. If you've ever put hydrogen peroxide on you, you know it bubbles up. It's because we have enzymes in our tissue that reacts with the hydrogen peroxide to form oxygen. And that bubbling is oxygen gas, and it's going to dislodge any bacteria that are in the wound. Our body makes a lot of toxic compounds, and they must be neutralized, and that's what peroxisomes do. Lysosomes contain digestive enzymes. You can think of it as the recycling center or the stomach of the cell. Microvilli are membrane extensions in areas that a lot of absorption is going to occur. So we'll see microvilli in parts of the digestive tract. The Golgi apparatus is the shipping department, and it modifies and packages proteins for export. The nucleus contains the DNA, which has the information to make proteins. It's surrounded by a nuclear envelope that is loaded with pores. Things need to get in and out of that nucleus. The endoplasmic reticulum is a series of channels to synthesize and transport substances. So the rough ER synthesizes. It's rough because it has a lot of ribosomes on it, and the ribosomes make proteins. Proteins enter the ER, and they are modified and transported. Smooth ER synthesizes lipids and carbs. Ribosomes are the protein factories. Mitochondria produce the ATP. They're the cell's powerhouse. And the cytoskeleton is an internal network of protein filaments that maintains some structure in the cell and also allows substances to be transported within the cell. Be able to recognize, identify, and determine the function of each of these different organelles. More review. Plasma membrane is a phospholipid bilayer with the hydrophilic heads at the surface, hydrophobic tails sandwiched on the inside. So it's like a butter sandwich where the hydrophilic heads are the butter, and in between will be the fatty substances, the hydrophobic tails. It's selectively permeable. That means the membrane is what chooses what is going to enter and leave the cell. Integral proteins are just proteins within the membrane. They may contain openings to allow substances to cross. And outside the cell is this carbohydrate layer. And it has a variety of functions: binding to other cells, cell recognition, lubrication. And here's a picture of all of those components of the cell membrane. The cytoskeleton is the cell's skeleton. It is just a framework of protein filaments for support, strength, and here are all the components of the cytoskeleton. Most important, you should recognize microfilaments, intermediate filaments, microtubules. You don't need to know all these different things in this chart. Recognize some of these terms as part of the cytoskeleton. The endoplasmic reticulum is tubes or chambers, and it's important for the synthesis and storage of proteins, lipids, and carbohydrates. Rough ER is for protein production because the ribosomes make the proteins. Smooth ER is for carbohydrates and lipids. So you can see here in this picture the rough ER with all those ribosomes, and it's just flattened sacs where the proteins are produced on the outer surface, enter, and then can travel. They're transported and modified within the rough ER. The mitochondria, you don't need to know details of the structure of it, but the point of the mitochondria is to take our nutrients that we eat and convert them to ATP, which is the kind of food the cells need. So the number of mitochondria per cell varies. So obviously a cell that has a lot of energy needs, like a muscle cell, will have thousands of mitochondria. This process of conversion of our nutrients into ATP is called aerobic metabolism or cellular respiration. And it's called cellular respiration because oxygen, that's why we breathe oxygen. That oxygen we breathe in is needed by the mitochondria to completely break down our nutrients to form the maximum amount of ATP. So in the mitochondria, our nutrients are going to go in, and oxygen, where's our oxygen, is going to go in. Here's oxygen going in, and the gas that's produced is CO2 from the breakdown of our nutrients, and ATP. So that ATP is going to leave the mitochondria and go throughout the cell wherever it's needed to provide energy. The Golgi apparatus is going to receive transport vesicles from the rough ER. So here's our rough ER down at the bottom of the slide, and it's going to form vesicles in which, let's say, protein. Let's say this is a pancreatic cell, and this is producing insulin for transport. The insulin will be modified and collected and transported in this transport vesicle to the Golgi apparatus, and there it's going to be further collected and modified for transport from the body. So this is our Golgi apparatus in purple, forming these transport vesicles of the insulin, which is going to be secreted from the cell. And what actually happens is that the membrane of this little vesicle comes over to the cell membrane and fuses with it, opening the products to the outside. And there's a beautiful micrograph of that process taking place here, where the product is released from the cell. The Golgi apparatus is also going to renew the membrane, and it also produces the lysosomes that contain digestive enzymes. So again, the lysosome has three basic functions: fuses with other organelles and digests old organelles. It can fuse with a vesicle from outside the cell and digest what's ever inside. For example, bacteria. If it's a white blood cell, the lysosomes of the white blood cell will fuse with the bacterial vesicle and digest it. It will also be responsible for destroying the cell when it is injured beyond repair. You can just pause this to look at each part of this diagram. The nucleus is the biggest organelle, double-layered membrane, lots of pores to allow substances in and out. Which organelle is involved in protein synthesis? Ribosome. Which of the following organelles is not separated from the cytosol by a membrane? Oh, I told you not to worry about those membrane and non-membranous, but which of these here does not have a membrane? It is the centriole. Which of the following will be prominent in a cell that is actively manufacturing ribosomes? It's making a lot of ribosomes. Well, ribosomes are made in the nucleus, in the little nucleus, the nucleolus. Which of the following would be densely concentrated in a cell that secretes digestive enzymes? The rough ER to produce the enzymes, the Golgi apparatus to put it into a vesicle, and the secretory vesicles. Digestive enzymes are proteins. Many neurons cannot divide. What organelles will they lack? They will lack the centriole. The nucleolus is in this picture here. The nucleus has a blue membrane, and there's a darker purple nucleolus dead center here. Won't always be dead center, but that's where the ribosomes are produced. And you can see all these nuclear pores. Ribosomes have to leave the nucleus to go outside to make proteins. And there's the nucleolus. And also this shows very well all these nuclear pores on the nucleus. DNA and chromatin are in the nucleus, and DNA has the information to make proteins. Chromatin is it's the stretched out, loose, uncoiled form of DNA, like cooked spaghetti. Chromosomes, on the other hand, are formed when that chromatin coils and coils and coils to form a thicker, denser, smaller body called a chromosome. So here you can see this chromatin. In the nucleus, it just looks like, it looks granular maybe, but it's all long, long strands of DNA. And here's the chromosome. This one's replicated during the interphase of the cell cycle, and it's held together by the centromere. It's supercoiled.